So this work is titled Towards Cybernetic Modeling of Biological Processes in Mammalian Systems, and the focus is on lipid metabolism in the mouse macrophage cell. This work is done through Purdue University under the mentorship of Dr. Doraiswamy Ramakrishna in collaboration with Dr. Shankar Subramaniam's group at UCSD. So to preface the work, I want to first talk about the context. The ability of a cell to process and respond to its surrounding environment and in turn adapt in that space is regulated by a complex system of signals that relay information from outside of the cell to its nucleus. Signaling networks are inherently very complex and require a systems level approach to gain insight into the dynamics and connectivity that influence one another. So multiple layers of cellular regulation are actually required to deconstruct a network and its role within the organism. There are engineering methodologies that are critical for a quantitative understanding of physiological mechanisms in both normal and disease states. Mathematical modeling of complex biological systems is typically done in one of two ways. More recently, a black box approach like machine learning is very common. The availability of a vast amount of biological data has allowed researchers to train these models and then predict outcomes of various diseases. But understanding the inherent mechanism is not possible with this approach. An open box detailed mechanistic pathway model might actually be better, but lack of high resolution knowledge about the key players in the model can actually deter this effort. For example, a large amount of knowledge is necessary in order to understand how the gene expression occurs and why certain genes are expressed at a particular time. The Ramakrishna group has developed an elegant approach called the cybernetic model to resolve the limitations that I've just mentioned. A cybernetic approach provides a framework to model these complex interactions through the implicit accounting of regulatory mechanisms, assuming some biological goal. So the premise underlying the cybernetic framework is that the regulatory processes affecting metabolism can be mathematically formulated as a cybernetic objective through the U and V variables that constrain the network to achieve a specified biological goal. The goal-oriented control policies of cybernetic models have been used to predict metabolic phenomena ranging from complex substrate uptake patterns and dynamic metabolic flux distributions to the behavior of gene knockout strains. However, this has been done almost exclusively in bacterial systems. So regulation of metabolism in mammalian cells is achieved through a complex interplay between cellular signaling and metabolic reactions, as well as transcriptional changes. So through this work, we've established a framework for cybernetic modeling in the context of mammalian systems. One complex network that we explore is our immune system, and specifically the role of the inflammatory response. Inflammation is a key player in the progression of multiple disease states. Chronic inflammatory diseases are the most significant cause of death in the world and is associated with cancer, heart disease, arthritis, and diabetes. It's also a key player in acute inflammatory scenarios such as infections, sepsis, and most recently COVID-19. So in this talks, I'll specifically address acute inflammation. Regulation is a key part of the inflammatory response, and it is the body's natural and essential defense mechanism responsible for combating antigens, restoring homeostasis, and repairing tissue damage. The body has a two-line defense system that works against pathogens or germs that make us sick. Pathogens include bacteria, viruses, toxins, parasites, and fungi. And the skin shown in this image is actually the largest organ of our body. It acts as a physical barrier between invaders and our body. The body's second line of defense is the immune system, and it triggers an inflammatory response when there are foreign invaders that are necessary to fight off. We will focus on a particular cell type, the macrophage cell. So macrophage cells act as our first responders to a site of inflammation. And extensive studies have been done to mimic the physiological response of bacterial infection on macrophage cells through exposures of factors like lipopolysaccharide, or LPS, denoted in this image, which is actually found on the surface of bacterial cells, and an increased level of adenosine triphosphate, which acts as a danger signal in response to cell death in the surrounding environment. Both of these initiate a cascade of events, one of which acts as the activation for the COX enzyme. We consider specifically lipid metabolism starting from arachidonic acid because the lipid molecules of inflammation are the prostaglandins, and they're involved in transcription factors and regulation that give rise to cytokine release. And cytokines are small molecules that function in the signaling or communication between cells. One representative cytokine is TNF alpha, which is well established for its role in many pain models. So this is a detailed network for the depiction of LPS stimulated, or TLR4 pathway for lipid metabolism and the signaling. The metabolites are colored according to their lipid categories, which are shown in this bottom corner. And as you can see, the network is actually fairly complex. So we actually focus on one aspect of this, the eicosanoids. And the black lines represent lipid metabolism, the red lines represent signaling pathways. And metabolites are represented in rectangular boxes, and then the enzymes in oval boxes. And the measured and unmeasured metabolites are also differentiated based on thick and thin borders. So the branches of the network that we focus on for this modeling framework are actually further simplified down to this image here. And what you see is we have the leukotrienes and the prostaglandins. The prostaglandins have been found to be responsible for initiating acute inflammation and mediating pain during the inflammatory response, whereas the leukotrienes are known to increase the expression of adhesion proteins and promote cell motility. So the structure of kinetics for this reaction network is arranged into segments. The first is emphasized in this gray here, and you can see that it's described uh, by the conversion of arachidonic acid into PGH2 using simple linear kinetics. The kinetics of this reaction is modified or modeled as three separate mechanisms. The first includes the metabolic flux, and then we have a degradation term, and of course there's the consumption rate to downstream metabolic products. And the metabolite flux is modeled in three parts. We have the basal rate of synthesis, and then the generation due to ATP and due to KLA stimulation. The other segment of this model employs a cybernetic framework to capture regulation between the different metabolic options. So similar to before, we write out the kinetic balance for the metabolites. Again, Ri kinetic represents the kinetic rate of the reaction, and Vi is the cybernetic variable controlling enzyme activity. And we now have what's termed the regulated rate. And of course, we also include the degradation as well as downstream metabolite consumption rates. And now we write out a kinetic balance equation for the enzyme. So alpha is the constitutive enzyme level, and this second term is the inducible enzyme level. And of course, we've got enzyme degradation. UI is the second cybernetic variable that regulates induction of enzyme synthesis. So the cybernetic control variables U and V will be discussed shortly. This highlight here is for the branches of the network that I'm going to focus on for this illustration of defining the cybernetic goal and calculating the U and V. So here we define the cybernetic goal for the network where the prostaglandins are well characterized for their role in the inflammatory response. So we're looking at the regulation of prostaglandin synthesis in relation to TNF alpha, which is a representative marker of inflammation, and in this case is our model's overall goal. So to quantify the relationship between the prostaglandins and TNF alpha, we used a simple linear relationship of the two using time series data. For the prostaglandins, we used existing metabolite data, and then for the TNF alpha, we used proteomic data. To determine the contribution of each PGI pathway that leads to TNF alpha production, we define associated weights WI of each pathway. So this return on investment rho is used to calculate the cybernetic control variables, which regulate the enzymatic activity and synthesis of the cell. So the cybernetic variables compare this return on investment rho for each pathway and then invest in enzyme production accordingly. Again, the cybernetic control variables U and V are computed from the matching and proportional laws, which you see here. Before we complete the discussion from earlier, where we were breaking up the AICHE network into parts, how do we actually obtain our cybernetic model? So here we formulate the cybernetic model in this box here using kinetic equations with the addition of cybernetic control variables. And then the U and V is each computed from the reaction fluxes at a given branch point. The dynamic model consisting of kinetic mass balance equations is solved numerically in this box here. And the sum of the squares of the error between the simulated metabolite concentrations and the experimentally measured concentrations is minimized in order to estimate our parameter set P. And now this brings me to the actual data that we have available to us. So this comes from the lipid maps database built through the Subramanian lab. And we use time series data from the bone marrow derived macrophage cells of mice taken from samples at 8 different time points that's shown on this timeline. And the data we have includes both lipidomic and transcriptomic data collected from mass spectrometry measurements as well as microarray data. And the different experimental conditions include a control case, a treatment with ATP, and then we have a combined treatment of ATP that's following a 4-hour pre-treatment of catiolipid A. Now, catiolipid A is symbolizes KLA in this, and KLA is a chemically defined substructure of lipopolysaccharide, which I mentioned earlier is actually the bacterial molecule that specifically induces an inflammatory response in macrophage cells. So regulation of arachidonic acid is fairly complex, and because we don't have any data on the upper part of the network, we simply used arachidonic acid as the initial input to the model and only simulated the downstream products. And you can see here that the mean experimental data, which is shown in circles with the associated standard error of the mean, are plotted from from the replicate experiments for the ATP stimulated case, which are in green, and the control case, which are in red. The simulation results are shown in solid green and solid red lines. And visually, you can see that our model effectively describes the conversion of arachidonic acid into the downstream prostaglandin products. We went on to test the validity of our model using a third independent data set, and we used the parameter values determined from our previous simulation to be able to predict the results of the combined KLA primed KDO, KLA primed and ATP stimulated BMDM cells. Again, the mean experimental data are shown in circles for the treatment case, which is magenta, and the control case, which is red, and the prediction yields of a very good fit to the experimental data, which further validates our model. But we did also perform some statistical tests to determine quantitatively the effective fit of our model. And we did this using a goodness of fit F test for fitted ATP stimulated data and predicted KLA plus ATP stimulated cases. And the F values that are smaller than a specific threshold and indicate that the fit error is statistically smaller than the experimental error, which is what we see in our model. And the eicosanoid model robustness was evaluated by performing a parametric sensitivity analysis in which the parameter was individually varied by plus or minus twofold of the original optimized value. So the slope of each parameter and metabolite sensitivity curve was calculated to evaluate that sensitivity, and a heat map of that of those slopes was generated, which I show in the image here. So small to moderate sensitivities in most of the parameters were observed. And based on these results, our model of eicosanoid metabolism is shown to be robust with respect to parametric perturbations. And then finally, we wanted to confirm the reliability of calculated rate constants to effective rate constants for those found in literature. So for example, we looked at COX, PGDS, and PGES. And then in our case, our computed range for PTGS indicate that the range that we report includes the reported value in the literature. And the finding that the flux distribution between pathways is comparable to previous reports has an important implication for the validity of our computational model. So this work has laid the groundwork for application of cybernetic modeling in complex mammalian systems, and the insight into the metabolic goals of multicellular systems allows for a more complete understanding of how the states of these cells evolve over time. So this insight can provide us the necessary knowledge to develop a greater understanding of dysregulation of the immune response and in the development of therapeutics for inflammatory diseases. With that, I'd like to acknowledge my research advisor, Dr. Doraiswamy Ramkrishna, as well as our collaborator at UCSD, Professor Shankar Subramaniam. Thank you for attending this talk, and feel free to drop any questions that you might have in the chat.